So should I start the presentation?
I'll introduce you in just 1 minute.
And after that?
Yeah.
I should...
Okay, so hi, I know people will be logging on as I go over this intro. But just in the first place, hello and welcome to Webinar Wednesday. So I'm Lee Munger, SEB's Communications and Membership Assistant, and we're really excited to have Shaili, Johri with us today to present taking advantage of the omics revolution for monitoring and conservation of wildlife populations. So a couple of ground rules first. We have you all on mute to cut out background noise, but would still love to hear any questions that you have. So if you'd please type them into the chat box, Shaili will get to them as many as she can at the end. So the presentations usually last between 30 to 50 minutes. And then that time before 11 will be for questions. And if we don't get to them, feel free to send me an email, kmunger@combio.org, and I'll pass those questions on. So just a little overview of the presentation. Shaili will present research on apex predator populations, wolves, sharks, and killer whales with respect to their genomic, transcriptomic, and microbiome profiling. Shaili will also discuss the use of these measures in understanding population size, structure, distribution, health, species interactions, and the effects of pollution on wildlife. Last, implications of these data in conservation and management will be discussed. Just a little bit about Shaili. She's a geneticist with a research focus in conservation genomics of wildlife populations. She completed her BS and MS in India and moved to the United States for her PhD in genetics at the University of Utah, Salt Lake City. She's currently pursuing her postdoctorate in population genomics of sharks at the Hopkins Marine Station, Stanford University. She works in interdisciplinary research areas to guide conservation policy. Her research career spans projects relating to conservation of tigers in Western India, wolves in Northwest USA, killer whales in the Pacific Northwest, and now sharks and rays in the Pacific and Indian Oceans. Her role as a project lead involves developing genomic tools to assist with biodiversity assessments, health monitoring, wildlife forensics, and fisheries management. In parallel to research, she works in close collaboration with fishing communities to develop science-based conservation policies through cross-sector collaborations, outreach, and education. She also engages in capacity building for genomic methods within wildlife research managers in the US and abroad. So now I'd like to hand it over and get to this great presentation.
Thank you, Lee, for organizing and for the introduction. And good morning, good afternoon, good evening, depending on where all of you are. I'll just start the presentation here. All right, so today I'm going to talk to you about using omics, which includes genomics, transcriptomics, and metagenomics for wildlife monitoring and conservation. And I think I might have just been a little bit overambitious when writing the abstract. I don't think I can cover all of the research projects that I've done, but I will talk about my recent research, which deals with shark conservation. And I can try and talk about a little bit if there are questions about the other stuff, if we have time in the end. So with that. So just for some of you who are early career researchers, I did my BS and MS which was focused on mostly microbiology and biochemistry in India. And then I did my PhD in genetics at the University of Utah, Salt Lake City. And there I mostly worked with Drosophila and C. elegans, which are fabulous genetic model organisms. And it was during my PhD that I kind of realized that I can only work in wildlife conservation for the rest of my scientific career, or I can't do science anyway else. So that's when I switched to using genomics and genetic tools to study wildlife and for their conservation. And that research kind of spans a little bit with tigers, now with sharks, previously with wolves and killer whales. So I've been lucky enough to work with apex predators. But when I was growing up and I had always been interested in wildlife, and wildlife conservation. Little did I realize that wildlife conservation is more about human rehabilitation, and then second about monitoring and conservation of wildlife populations. So in every project that I've realized that I'm somehow studying interactions of the wildlife populations with their human counterparts and trying to mitigate the situations that arise to keep both sides in balance. So just to briefly, so for example, when I worked at the Tiger Research and Conservation Trust, very briefly, my role was to figure out how do we figure out alternative sources of livelihood for humans that have relied on these forests for the longest time. But now, because of increase in the human population and decrease in habitats available to tigers, there's more and more conflict. With sharks, it's mostly about dealing with the illegal wildlife trade and trying to protect remaining populations. With wolves, which was my research project at the University of Washington, it was about figuring out where these recovering wolf populations are found so that we can reduce conflict of wolves with ranchers and other protected wildlife. And killer whales are one of the most polluted animals today because they're at the top of the food chain, and they accumulate, they bioaccumulate for a really long time. So there it was trying to figure out how pollution is affecting killer whales. So as you can see, all of them are somehow, all of this research is juxtaposed with human-wildlife interactions. Today, I'll, however, talk to you about one of these projects, which is conservation of sharks, and how I'm trying to use genomics for this purpose. So this research I pursued previously at the San Diego State University, and now I'm working at the Hopkins Marine Station with Barbara Block as part of Stanford University. So for most of us, our perception of sharks comes from this not so great movie, Jaws. I mean, I have nothing against films, but this particular film, I think, has done quite a bit of disservice. And it has casted sharks into this really negative picture. However, as opposed to that, sharks are really gentle giants for the most part, and they're not interested in humans. Here you can see this whale shark. This is a picture that I took down at the Ningaloo Reef in Western Australia, and you can see people swimming right next to it, and it is fine. And this has been the experience with most of the sharks that I have worked with or have heard other people work with. And this is not moving forward. Oh, there we go. In addition to that, sharks are at the top of the food chain. So they are apex predators. So they're really important for maintenance and management of our marine ecosystems. So for example, in coral reefs, as you can see on the left here, where sharks were removed from the system, there was an increase in mesopredators, which led to a decrease in the herbivore fish populations. And this led to an increase in algae. And when you have increased algal cover, there is less of a coral cover. So sharks are really important for the maintenance of coral reefs. Sharks are important for our own food security. So in the mid-Atlantic, where most of the big sharks, the apex predators, were taken out, there was, again, an increase in mesopredators, and that led to a collapse of the scallop industry. So, like I said, sharks are really important for the maintenance of marine ecosystems and for human food security. Sharks are also really important directly and indirectly for the economy. So like I said, if you don't have sharks, you don't have a good coral cover, and that affects tourism. of coral reefs. And there's also a lot of emerging shark tourism around sharks themselves. So in the Bahamas, a live shark is worth $250,000, whereas a dead shark is worth $50. So in addition to all of these benefits, sharks are also just very fascinating as a model system to study. They've been around for 500 million years approximately, so they have the ability to survive a lot. They are among the oldest surviving vertebrates today. So they have high evolutionary resilience, very long life histories, and which tells us that they should have a very good genome stability, immune function, and wound healing. So they're just a really interesting biological system to study. So that's why, because of all of these functions, we want to preserve sharks. However, the illegal wildlife trade really threatens sharks in race today. So approximately 300 million sharks are killed because they're targeted or caught as bycatch for the fin trade, or rays are caught for their gill rakers. Shark meat is now emerging as a very big product in demand. And then a lot of the liver oil, squalene, cartilage, etc., is used for human medication, and they're also found in human food. For example, a lot of fish and chips in the UK is actually shark and chips because they're just, the meat is much cheaper to get. So as a result of this, there has been an 80 to 90% decline in a lot of different species like the great white, the thresher tiger, the scalloped hammerhead, so on and so forth. When we look at where these decline in shark populations is concentrated, so this is a map from Dalby et al., and the yellow to red colorations show the concentration of threatened species, and you can say they're concentrated much around South America, Africa, and then around Asia. The gray to black colorations are showing you the landings share. So for example, India has one of the highest landings share for shark landings. And then the size of the fin shows you what is the contribution of these locations to the fin trade. And one additional information also in this graph which is not in the legend here, is that the data deficiency. So the reason it is also hard to preserve and conserve remaining shark populations is that there is a lot of data deficiency, meaning we don't know enough about the shark populations. And the data deficiency is also the highest around areas where there is the highest concentration of threatened species, which is Africa and Asia. So when we think about conservation and management of remaining populations, there are several things that we need to consider. One is to identify species targeted in the fin trade or in the wildlife trade, and see which species need to be prioritized for protection. Second is to understand species distribution, population structure, and size, so that we can figure out where remaining populations are and we can protect them. Next is identifying threats to the populations. And I would say one of the most important parts is to do stakeholder engagement, outreach, and education among the communities that engage in shark fishing and trade. So when we talk about the first issue, and let me just mention that all of these pieces of information come together in coming up with a conservation and management plan that is sustainable. So going back to the first part, where we need to identify species for wildlife trade, is the methods that I used. So most parts, most places, and most people. Sorry, that's not the right way to put it. So one of the most popular ways to do it is to use taxonomy, where you use morphological features to identify the shark species. But when a shark has been dismembered and cut into fins and meat, it's very hard to know what shark or ray species we're dealing with. So molecular identification, which is using DNA, becomes really important. And traditional methods for molecular identification require much infrastructure, and they're not accessible to in places where there is the most need, which is the red parts of the map that I've showed you previously from Dalvi et al. So one of my research goals was to kind of decentralize and decolonize the study of sharks. And I wanted to develop a method that could be used anywhere and by anybody. And that's where one of the devices that was recently developed by Oxford Nanopore Technologies in the UK kind of came handy. So this is like a handheld DNA sequencer in which you just put DNA and you plug it into your laptop, and you can be getting sequence data in real time, as opposed to sending tissue samples or DNA to a sequencing facility, which takes about 24 hours or more, depending on where you are. And very often people have to ship it out of town and overseas to get this analysis done. So I developed this method called genome skimming with the sequencer, which is called the MinION. Interesting names. So in this case, you take genomic DNA from a fin sample or whatever tissue you have at hand, and after about 10 minutes of preparation, you feed it into the sequencer. And that allows you to get very long sequences, including mitochondrial sequences, which are extremely conserved within species and differ between species, and are very useful in identification of the species. And because you have large parts of the genome, of the mitogenome available, there is less possibility of false identification. So you have very high accuracy. In addition to that, you also get nuclear sequences, as I'm showing on the right, which will eventually, with further analysis, allow you to understand population structure, size, and more about species biology. So this method was published last year in Scientific Reports. So we tested this method in India, which I mentioned to you has a very high landing share. So India is actually the second largest shark landing nation in the world. And within India, the northwestern province of Gujarat has the largest contributor to India's shark exports. So there I collaborated with fishery scientists shown here in the picture, Jitesh and Vishal, and this is my colleague Mike Dawn from the Dinsdale lab, who is now working in Australia. And we sampled fins that were being sold in the market in Gujarat, and these were slated for export. So at this point, they had been dismembered, and we had no idea what species it is. So we took some DNA samples, we took some tissue samples and ran it on the MinION sequencer and asked what species it is. And the process was like, the DNA extraction took about 3 hours, and then the preparation of the sample took about 10 minutes, and then we put it on the sequencer for about 48 hours, connected to a laptop as shown here in the picture. At the end of 48 hours, we got about 74,000 sequences, and the longest sequence was about 100,000 base pairs, which is really high if you compare to other sequencing methods where the maximum is about 300 base pairs. And then we got the mitochondrial and nuclear genome components. The accuracy of these sequences was at 99.8% consensus accuracy, and the GC coverage ranged from 29 to 60%. One of the undergraduates who is now a PhD candidate, Isabel, helped with the DNA extractions and analysis of this data. So what did we get? So when we take these 74,000 sequences and align them to already available mitochondrial sequence from the GenBank database of reference sequences, we found that 47 of these reads aligned to this mitochondrial reference, and we did that with a few different mitochondrial reference sequences, just to be sure that we were not biased. So what we found was that in the first 2 minutes of our sequencing run out of the 48 hours, we were able to get almost a complete length of the mitochondrial sequence. And within about 180 minutes, we were able to get almost all of the sequences that would allow us to get about a 20x coverage of the mitochondria. And this we can tell based on when the sequence was obtained from the sequencer, so there is a timestamp. So essentially within 2 minutes to 3 hours, we can actually get the mitochondrial sequence and determine the identity of the species that the tissue came from. So these 47 sequences gave us a complete mitochondrial genome. And when we did the phylogeny with help from Sam Fellows, who is also now a PhD candidate at UC Merced, we identified the species to be Calcareinus falciformis, which is a silky shark, and it's actually a CITES-listed species. So we had now identified illegal trade of the species because it is CITES-listed and it should not be traded without a permit. And like I mentioned, one of my goals was to decentralize and make this accessible to everybody. So we came up with 2 different methods of analysis for this data. One is that can be done quickly in the field using a localized database on the laptop. And it may not need internet. And the other one is where you do a more high throughput and extensive analysis, which would require more computational power and would be done in the lab. And we did this with help from Adrian Cantu, who is just about to finish his PhD at San Diego State University. So the field method can be used for quick identification and more for wildlife forensics and law enforcement purposes. And the lab method can be used for more extensive analysis like population genomics, so on and so forth. And in our paper, we have the entire pipeline for this also available through a Jupyter notebook. So you can use the programs, et cetera, and the commands that we used to do this analysis. So this was data that I just showed you from one species. But what does that mean for the overall wildlife forensics and wildlife conservation field? Excuse me. So I mentioned that the GC content varied from 29 to 60%. So one of the main hindrances to sequencing different kinds of DNA from different species is the GC content, and it can different sequencers can be limited in terms of what is the GC content or the range of the GC content that they can sequence. But we have now sequenced about 7 species of sharks and rays using the Menahan sequencer. And in that, we see a GC range of 29 to 60%, and that GC range includes a lot of other species as well. For example, the pangolins are at 37% GC content, so they could be easily sequenced with the sequencer. Elephants are at 39% GC content. Sharks and rays are at 42%. Rhinos are at about 51%. So that tells you that this method of genome skimming and sequencing on the Menahan sequencer can be used for a lot of the highly endangered and threatened species. I've also worked closely with the San Diego Conservation Research Institute, and where they're trying to revive the northern white rhino, which has gone extinct. And they're using a lot of genomic and transcriptomic tools. And there we were able to do some of the southern rhino sequencing for this project using the MinION. And we are now looking at doing some elephant ivory sequencing on the MinION to differentiate between legal and illegal ivory. So because of the versatility of this method, our article and some of our research was covered in National Geographic, which was really exciting. So that brings me to the next part of my research, which I'm currently pursuing. So once we had the species identification nailed, we next wanted to see if we can use the MinION sequencer for population studies, which involves population structure and size, but also species distribution. And this also needs to be made available to places of high threat because we need to really preserve remaining populations. So this project I'm pursuing in another part of the Indian Ocean, which is in the Chagos archipelago, which is this little, it's little here, but it's actually pretty big. So which is this part of the Indian Ocean, I think about 1,500 miles from the southern tip of India, approximately. So the Chagos Islands archipelago is the largest no-take marine reserve in the world. It consists of about 544,000 square kilometer area. It has about 55 islands and 300 seamounts and abyssal habitats. It also has the largest coral atoll on Earth. So even though this is a protected area, there is historically and recently there has been a high rate of illegal, unregulated, and unreported fishing, as it's called IUU fishing in the area. And the reason is because the area is so large, there is very little enforcement and patrolling of the area. And this is the case with most of our oceans, that the capacity to patrol and monitor these areas is really low. So what I'm showing you here in this figure from Ferretti et al. Who is a previous member of my current lab, is these yellow spots are the different illegal fishing vessels that were found over the years in the Chagos. And on the right here in the study by Tickler et al. 2018 as well, is these areas where a patrolling boat actually was able to catch illegal fishing in action. So as you can see, there is this spot, this, and this. So there are quite a few places where there is illegal fishing. And these are just the areas where they were actually caught in action during the patrolling. And when they examined what was on these boats that were doing IUU fishing, there was a large amount of sharks and rays. They also found a lot of manta rays, which is not being shown here. So essentially, sharks and rays are targeted in IUU fishing in the Chagos. And I think I didn't mention this earlier, is that sharks have a really long life history, which means that they take a really long time to mature and they produce very few young. So any amount of pressure, fishing pressure, is extremely challenging for them to recover from. And this becomes really critical for a place like the Chagos, which is a coral reef, a protected coral reef, that if we are removing shark populations, it is really going to affect the health of the reef in addition to the rest of the marine ecosystem. So there are several species of sharks found in the Chagos, including the silky shark and some other pelagic species like the tiger shark. But two of the most common species found here are the gray reef shark and the silver-tip reef sharks. And what I'm showing you here is that they are much below their carrying capacity, both of these populations. So the carrying capacity is shown by this straight bars, green and blue respectively, for gray reefs and silver tips. And over the years, these populations have declined quite a bit, mostly, as we understand, because of IUU fishing. So what do we do? So the project that I'm pursuing aims to identify and protect current strongholds. So for that, our collaborators at ZSL, they are collecting the Zoological Society of London and our lab. We've collected DNA from different sites in the Chagos, and then we filter this DNA, and we collect water from different sites in the Chagos, and then we filter this water to isolate cells or DNA that has been shared by species that have been in the area. Then this DNA is sequenced, and from this sequence, we try to match the DNA sequences obtained from the water to an available database of reference sequences. And from here, we identify the species, and then this tells us which species are found where in the Chagos Islands. However, one of the challenges here was that our two most common species, the gray reef and the silver-tip sharks, had very similar DNA sequences, and their mitochondrial genomes were not yet available. So from the available sequences in the database, it was very hard to determine which species it is. So we needed the mitochondrial DNA sequence for these, so that we could compare the entire mitochondrial DNA sequence and then come up with the region that differed between the two species, and then use that to identify the species from our water DNA samples, which are called environmental DNA or eDNA samples. So we use the MinION again to sequence the mitochondrial DNA from both of these species using tissue samples. And that gave us the entire mitochondrial sequence for both of these species. And now we are aligning these sequences with each other to determine areas of genetic difference, and then use that to differentiating DNA piece to identify the species from the DNA that we have collected. So this is another application of the genome skimming method for determining species distribution. We are just starting to get to determining population structure of these two species in the Chagos Islands. So for example, what I would like to know is that among all of these different islands shown here in this slide, if we have sharks that were collected close to these different islands or atolls, do they differ in terms of the population structure? And are there genetic differences that we can identify between these subpopulations? So for example, here I'm showing you just a couple samples that we took. So there was, at the bottom of this slide, there is two silver-tip sharks that were collected about 6 kilometers apart from each other. And when we compared their mitochondrial genomes, they were very similar, with like a couple base pairs of difference. However, when we collected gray reef sharks from about at a 60 kilometer distance from each other, from two different islands in the Chagos, we found that there is much of a difference in the mitochondrial sequence. We haven't looked at the nuclear sequence yet. We're still analyzing this data, but that tells us that there might be some population structure, and we might be able to use these differences in the genomes of these subpopulations to see who might get targeted. And I can explain that a little bit further on this slide. So, as I said, there is illegal fishing going on everywhere, and a lot of these boats have been identified to be coming from India, Sri Lanka, and Africa, South Africa, possibly in some cases. So, if we were able to identify genetic differences between the subpopulations, let's say, of the gray reef sharks, and then we compared these genetic signatures to gray reef sharks found in the markets of, let's say, India and Sri Lanka, we might be able to identify poaching hotspots around the Chagos where there is high degrees of illegal fishing, and then we might be able to focus law enforcement in these areas. So that's our long-term goal. And so two goals of the study here. One is to identify current strongholds, and then to identify poaching hotspots, and then use both of these tools to protect remaining populations. So this is ongoing work. And that brings me to the last part of my talk, is stakeholder engagement, outreach, and education. And this, I would say, is one of the most important aspects of the work we do, because more often than not, the science is really important in determining where the species are found, what is the population size like. But if we don't rehabilitate and don't inform the fishing communities that are engaged in conservation of sharks, in the fishing of sharks, and the communities that rely on these fisheries for their livelihoods, we are not going to be able to come up with a sustainable solution. So that's where we are also focused. So much of this work was done by another student of mine. Her name is Anjani Tiwari, and she's currently based in India. So when we collect these samples for molecular identification and biodiversity surveys in India, we were also surveying the fishing communities and working in close collaboration with them to be able to get these samples. But what we also did was to get an understanding of what are the knowledge gaps, or what is the information we can get from them, and what are the gaps that we need to fill in terms of conservation and management of the sharks. So in our surveys, there were some questions we asked, and one of the key things was, what is the support of the community for conservation? And what we found is that about 70% of the communities were happy to help us in conservation, because sharks were not a big part of their fishing livelihoods. And that's because most of the sharks are caught as bycatch. And while that bycatch is enough to damage the shark populations, it's actually not a big part of the sustenance of these communities. The second was, one of the other key questions was, what is their knowledge of the ecological role of sharks? And what we found was that about 90% of the fishing communities did not know that sharks and rays as apex predators are really important to maintenance of marine ecosystems and to healthy commercial fisheries. So when we exchanged this information with them, there was heightened support for shark conservation. And most of these fishing communities, which I'm talking about 500 people, and most of these people had about 100 trawler boats of their own. So you can just understand the extent of fishing that happens, and how important it is to take this knowledge to these folks, and to involve them in any of the conservation and management programs. So most of these fishermen and fisherwomen told us that nobody had exchanged this information with them in the last 30 years of their professional lives. So, and a lot of them told us that they're already noticing a decline of about 20 to 40% in their shark catches, and also in the catch of other commercial fisheries. So I think if we engage at a regular basis with these communities and establish partnerships with them, it would make a significant difference in developing sustainable conservation programs. So these are results from some of the surveys we did, but the other thing that we do is once we have collected the data and we have done our analysis, we actually take it back to these communities. So here you can see Anjani sharing results from some of her studies and in the poster that she presented at the last IMCC with the fisherwomen here, and they are really excited to be part of this. And this is how we also establish partnerships with them. This is me trying to talk to fisheries students at the college, at the fisheries college, and kind of recruit students from these fishing communities to be involved in policy making and fisheries management for the next generation. We also do this with at primary and higher secondary schools where kids from the fishing communities go to, and most of these kids have no idea that there are sharks in the waters that they live next to. So, and some of the questions and the engagement that we get from these students, both at the school and the university level, is just amazing. These two are posters that we made in the local language to kind of explain the ecological role of sharks as top predators. So when you have hammerheads that are happy, there is a healthy ecosystem, and there is fisheries, there's good fishery supporting shrimp, etc. And then on the right here, we have poaching of sharks for the fin trade, and that leads to a collapse of the shrimp industry. And then here we are showing a comparative life history of tuna as opposed to sharks and rays, where we're showing that a tuna or other teleos fish, for example, produce thousands of babies in a very short amount of time, in a short generation time, whereas chondrichthyes or elasmobranchs have a very high generation rate, very long maturity time of about 10 years, and then they produce very few young. And a lot of this work and this material was prepared by Anissa and Isabella, who are undergraduates going on to master's and PhD programs at San Diego State. So that brings me to my conclusions and future directions. So far, as I mentioned, we have sequenced about 7 to 8 species, including sharks, rays, and rhinos. And we are going on to sequencing probably elephants in the next year or so for wildlife forensics. We're just getting started with our population genetic studies. Again, for wildlife forensics, but also for conservation and management. And this might have implications for monitoring of subsidies that are sustainable and being used in a fair manner, and also monitoring our food quality, determining bycatch rates in fisheries and the population health of the bycatch industry, of the bycatch species, and then again in law enforcement and conservation. We continue to engage in outreach and education, as I just mentioned in the last few slides. And we are also much involved with capacity building. So as an example, there is going to be a sequencing at sea workshop at the next IMCC in Kiel, Germany. So I would invite you all to participate in that if you're going to be at IMCC. So with that, I would like to thank all of my collaborators. And these are collaborators about whose work I talked about today, but also whom I didn't get to talk about today. And with that, I'll take any questions. So one of the questions is, how much does a MinION cost? And the MinION costs about $1,000. And the first time you get it, it includes 2 flow cells. And if you order the flow cell outside later on, it costs about $400 to $800, depending on how many you get. But there's also a smaller flow cell that is available, and that's about $90. So depending on your needs, you can use a flow cell accordingly. So I don't see that there are a lot of questions that Lee hasn't addressed.
There's one more from Monica. Can the MinION detect multiple species in one sample? For example, if collection biological samples from a carcass, can it distinguish both the carcass species as well as predator scavenger species that have left saliva on a carcass?
Yeah, so there are about 2,000 pores on the flow cell, and I think a lot fewer on the smaller flow cell that I mentioned. So you could analyze these sequences separately, and you would be able to identify all of the sequences that are, all of the species that are present in that sample. You could also run multiple samples in parallel by using barcodes. So you could barcode each sample up to 12 in a flow cell and run 12 samples in parallel as well. I don't know. I don't know if that answers Monica's question.
Great, thank you. And then it looks like there's 2 more, one from Tracy and one from Junit, if you can see those.
Yeah, I don't see them.
Okay, I'll just read them for you real quick. From Tracy, how widespread has the MinION been used by enforcement agencies, and are they finding this tool useful in meeting their needs?
So I would say that the MinION is actually not being used extensively by wildlife enforcement. I think there's just now there's going to be interest, and people are trying to validate and train people to use this. But yeah, so I think there's just the use is just now starting to happen. I don't think it's being used very extensively just yet.
Okay, thanks. And then, is there funding available to join the training courses you do?
I think it would have to be on a case-by-case basis. I think the course that I'm running, the workshop that I'm running at IMCC is going to be very minimally priced. So it's almost negligible. So I think the cost would be for you to attend and travel to IMCC. Yeah, because most of the reagents and stuff is already paid for. So these are essentially going to be free workshops, other than the cost for securing the space at the conference venue.
Okay, great, thank you. And then Vicky would like to know, do you need to isolate the saliva or can the sample be mixed?
Yeah, I think can you elaborate on that question a bit, Vicky, if you can hear me?
We'll come back to that. We'll come back to that if Vicky responds.
So I don't think you don't need to isolate the saliva, if I understand. You would just take whatever DNA is available at the site of the bite or whatever is and extract the DNA and then load it into the MinION sequencer. And you should be able to get all of the DNA sequences deposited by all of the species there. And you can match all of these sequences to a reference database and you should know what species there are.
Okay, great. And then Dale would like to know, what are the database requirements and costs for remote field work without internet connection?
So depending on what your project needs are, you can just download these sequences from GenBank and have it on your computer. And if you are if you have a lot of experience with the bioinformatics needed, you can just have it running in your terminal or whatever program you use. But oftentimes, a lot of people use Genius, which is actually a paid program, and that is more, it's a more user-friendly interface where you don't need a lot of bioinformatics expertise. And you can just have that local database loaded into this program called Genius and match it up. But I think there is also Mega and other programs that are free. So there is a way to just do this for no cost, actually.
Great, thank you. And then Li Chen wants to know how to estimate the population size using DNA data, given potential bias in sequencing and decoding.
So I think it's pretty well established how you can do population size estimations from population genomics from sequence data. So I would refer you to read these papers, how they do it. And depending on your study questions, you would be targeting the DNA to be sequenced and then analyzing it. And there are always precautions taken to remove certain SNPs from your analysis when you're doing population estimates. So I think there are a lot of studies about this.
Great, thanks. And then Monica had another question. I am also curious specifically what research you are planning with ELES. Is it similar to the work of Sam Wasser at University of Washington?
What is ELES?
I'm not sure. Monica, if you could elaborate, that would be great. Elephants.
No, so I think Sam has already done some great work on that. We are just looking at, and this is still a project in development where we're trying to, so for example, there is Asian ivory, Asian elephant ivory is legal to be sold in some parts of Asia, whereas African ivory is not legal to be sold. But often these two are mixed and laundered in the market. For example, in places like Malaysia. And there we are just trying to figure out a way to streamline identification of these two different ivories by law enforcement so that they can then figure out which ivory is legally being sold and which is not.
Great. And then one more, do you find this a practical method in developing countries with regards to the costs?
So I think the cost at upfront might seem high, but just the amount of data that you can get from this method is pretty high. And there is a continual effort from Oxford Nanopore and from myself as well to figure out ways to reduce the cost. So I think it is absolutely doable. And we have, there are just a lot of collaborators from developing countries who have approached me and are very interested in pursuing this.
Okay. Awesome. Does anybody have any other questions? We probably have time for one or two more. And if not, then we can go ahead and wrap up. Okay, one more. How clean are the environmental samples? How clean should they be to identify species using the MinION?
So I think the MinION is just a sequencer, and so you would essentially take all of the precautions that you usually take with any environmental sample. So, for example, when we do environmental DNA sampling, we try to not introduce any human DNA, but I think it's hard. So we take DNA in sterile bottles, and then we pass it through a filter, which is also sterile, through vacuum filtration. And when we are doing this, we have a filtration blank to make sure that that any contamination introduced should also be in the blank. And then after that, we extract the DNA, and then we are amplifying a small part of the DNA for species identification. So the answer to that would be, I think you would just take the same precautions as you take for any other DNA sequencing.
Great. And then we have one last question from Vicky. Is anyone helping nonprofits and regional and national agencies to get together to share costs for MinION materials?
So I'm working with a couple NGOs to incorporate the use of the MinION in, for example, fisheries monitoring and management to determine what species are caught and if it's actually being done legally. Regarding the funding, I think it depends on a case-by-case basis. So the NGO would have to come up with their own funding. I think if you're first-time users, and then some of the costs are subsidized by Oxford Nanopore, like it's cheaper to buy your first kit than subsequent kits. But I don't know of a conscious effort where somebody is actually sponsoring this. But the good news is that it is becoming more and more part of mainstream use. For example, I know NOAA is also starting to use this quite a bit.
Okay. Great. Well, thank you everyone for attending the webinar, and thank you so much, Shaili, for this presentation. Just so everybody knows, this presentation will be available for replay through Webinar Jam immediately. I think you'll get an email with that link, and I will also be posting it on our Vimeo channel. It's just Society for Conservation Biology Vimeo. So thanks again, everyone, and I hope you have a great day.
Thank you all. Thanks, Lee.